One of the most important neurobiological properties of the action potential is the all or none principle, which precisely defines how a neuron responds to stimuli. It means that an action potential is generated only when membrane depolarization reaches the threshold value. If the level of excitation remains below this threshold, the neuron does not respond at all and no nerve impulse is produced. From a neurobiological perspective, this threshold corresponds to the moment of mass opening of voltage-gated sodium channels, which initiate an avalanche-like influx of Na plus ions. The all or none principle also means that the action potential always has the same form. It occurs in a single, invariant shape with a constant amplitude, independent of the strength of the triggering stimulus. A stronger stimulus does not increase the height of the action potential, but can only affect the frequency of its generation. Importantly, the amplitude of the action potential remains unchanged along the entire length of the nerve fiber, from the axon hillock to the axon terminals. This is made possible by active regeneration of the signal in successive segments of the membrane, ensuring reliable and precise transmission of information in the nervous system. The fact that the amplitude of the action potential is independent of stimulus strength necessitates the existence of another mechanism for information encoding. The nervous system solves this problem by encoding stimulus intensity in the frequency of action potentials. A stronger stimulus does not produce a larger action potential, but leads to more frequent crossing of the excitation threshold, and thus to an increased firing rate of the neuron. The higher the frequency of impulses, the stronger the signal transmitted to subsequent elements of the neuronal network. Additionally, in some sensory and motor systems, stimulus intensity is also encoded through the recruitment of a greater number of nerve fibers. As stimulus strength increases, additional neurons or neural units that were previously inactive become activated. The combination of frequency coding and spatial recruitment allows the nervous system to precisely represent a wide range of stimulus intensities, despite the fact that each individual action potential always has the same amplitude. The unidirectional conduction of the action potential is a direct consequence of the neurobiological properties of voltage-gated sodium channels. In the region of the membrane through which the impulse has already passed, Na plus channels, after a brief period of opening, enter an inactivated state. In this state, they cannot reopen even if the membrane becomes depolarized again, creating the so-called absolute refractory period. This means that the signal cannot travel backward into the region that has just been excited. At the site where the impulse is currently located, sodium channels are in the open state, allowing a rapid influx of sodium plus cations into the cell. This influx causes strong depolarization and the spread of positive charge along the membrane in both directions. However, only in one direction can the impulse continue to propagate. In the region located ahead of the excitation wave, sodium channels remain closed but ready to open. When the depolarization wave reaches this area, these channels open, initiating the next action potential. The wave of sodium ions cannot re-excite the region located behind the impulse because the sodium channels there are inactivated. As a result, conduction is directional and orderly. In myelinated axons, this mechanism is further optimized by saltatory conduction. The action potential jumps between the nodes of Ranvier where there is a high density of sodium channels. This adaptation allows conduction velocities of up to hundreds of meters per second and represents one of the most efficient mechanisms of signal transmission in the nervous system. The action potential curve reflects a sequence of precisely synchronized neurobiological processes occurring in the neuronal membrane and consists of three main phases. The first is depolarization, which represents rapid excitation of the cell caused by the massive opening of voltage-gated sodium channels and the rapid influx of Na plus ions into the neuron. This results in a dynamic rise in membrane potential and a reversal of its polarity. This is followed by repolarization, the phase during which the membrane potential is restored toward its resting value. During this stage, sodium channels become inactivated while voltage-gated potassium channels open, leading to the efflux of K plus ions from the cell and a gradual decrease in membrane potential. However, this process does not stop exactly at the resting potential. During repolarization, an after-hyperpolarization occurs. a brief period during which the membrane potential falls below the resting level, to approximately minus 80 millivolts. This results from the temporarily increased permeability of the membrane to potassium ions. From a neurobiological perspective, after hyperpolarization is functionally important because it reduces neuronal excitability and determines the relative refractory period. The entire sequence of these three phases ensures not only the generation of the action potential, but also its unidirectional and controlled propagation along the nerve fiber. The typical shape of the action potential is a direct reflection of precisely regulated neurobiological processes occurring in the neuronal membrane. The process begins with a gradual rise in membrane potential resulting from the summation of electrotonic potentials generated in the dendrites and soma. When depolarization reaches the threshold value, usually around minus 55 millivolts, an avalanche-like mechanism is triggered involving the rapid opening of voltage-gated sodium channels. This leads to a rapid influx of sodium ions into the cell and a sharp rise in membrane potential to positive values, reaching approximately plus 30 millivolts. This forms the characteristic peak of the curve, known as the spike. After this maximum is reached, a critical switching of ionic mechanisms occurs. Sodium channels become inactivated, preventing further sodium plus influx, while voltage-gated potassium channels open simultaneously. The efflux of K plus ions from the cell leads to repolarization, or the gradual return of the membrane potential toward its resting value. During this process, a brief hyperpolarization occurs, during which the potential falls below the resting level. This phenomenon results from the fact that potassium channels remain open longer than sodium channels, causing prolonged efflux of positive ions from the neuron. From a neurobiological perspective, the entire shape of the action potential is determined by differences in the kinetics of sodium and potassium channels, their rates of activation, inactivation, and closure. This precise sequence of events ensures the stereotyped nature of the nerve impulse, its unidirectional propagation, and the reliability of information transmission in neuronal networks. Refractoriness is a key neurobiological property of the action potential because it defines the minimum possible interval between successive impulses and thus limits the maximum firing frequency of a neuron. This mechanism arises directly from the temporal properties of voltage-gated ion channels and ensures orderly, unidirectional signal conduction along the axon. Two successive refractory periods are distinguished, differing in their mechanisms and functional consequences. The absolute refractory period occurs during depolarization and early repolarization of the action potential. During this time, generating a new impulse is impossible regardless of stimulus strength, because voltage-gated sodium channels are in the inactivated state. Until they return to the closed state, the neuron remains completely unexcitable, representing an absolute limit on firing frequency. This is followed by the relative refractory period, which occurs during after hyperpolarization. In this phase, the membrane potential is lower than the resting potential, mainly due to the persistently increased permeability to potassium ions. Although some sodium channels have already regained the ability to activate, triggering an action potential requires a stronger than usual stimulus. Neurobiologically, the relative refractory period allows regulation of firing frequency and precise encoding of information, while simultaneously protecting neuronal networks from excessive excitation. Synaptic transmission is a precisely synchronized process that begins when an action potential reaches the presynaptic terminal. Depolarization of the synaptic bout-on membrane, caused by the influx of sodium cations, leads to the opening of voltage-gated calcium channels. The entry of calcium 2 plus ions into the presynaptic terminal constitutes the key signal that initiates a cascade of molecular events responsible for neurotransmitter release. High local calcium concentrations activate specialized sensor proteins such as synaptotagmin, as well as the SNARE fusion protein complex, which enables the rapid fusion of synaptic vesicles with the presynaptic membrane. As a result of this fusion, the neurotransmitter is released into the synaptic cleft, from where it diffuses toward the postsynaptic membrane. There, it binds to specific receptors, leading to the opening or closing of ion channels and the generation of a local change in membrane potential. Depending on the type of receptor and the ions involved, Either an excitatory or inhibitory postsynaptic potential, EPSP or IPSP, is generated. These are electrotonic potentials that undergo further summation in the dendrites and soma of the postsynaptic neuron. The entire process of synaptic transmission occurs on a millisecond timescale and is characterized by extraordinary temporal and spatial precision.